All right, great. Well, thank you so much for having me. And it's always fun to talk to really a diverse group of people. You know, I'm guessing many of you are in engineering, science.
Atmospheric sciences, math,
Okay,
I'm surprised sometimes.
and how much neuroscience background? Like, you know, we have brains and neurons. Okay, great. So please interrupt. I would rather get through a third of my slides and have you learn something than get through all of the slides and have you fall asleep like halfway through, because yeah. Okay, so I am a physicist and I work in neuroscience, and that may seem really odd, but perhaps I can explain, you know, a little bit of the perspective that I bring there. So as with almost all fields of science, life, whatever, we have had a complete revolution in the quantity of data that we can obtain from brains. And one way of looking at this is the number of neurons that you can record from simultaneously. So for perspective, the human brain has like 100 billion neurons. We're not at that scale yet, but if you were trying to, you know, look at neural activity 50 or 60 years ago, you might get to see one neuron at a time. And just imagine trying to learn something about brain function from looking at one neuron. That has been doubling every about 7.5 years or so since then. I added, even since this was published 10 years ago, there's a few more points on this plot. It's now possible to record from thousands, tens of thousands. Somebody has claimed 100,000 at, you know, some lower temporal resolution, but many, many neurons simultaneously. So what does this kind of data look like? This is an example. This is one way that we often represent neural activity. And so this plot, as you, along the x-axis is time, and that bar right, okay, good, that shows up. This bar here is a minute, and then each row is a single neuron. This is about 500 neurons, so there's, you know, 3,000 or so here. What you can see from this, all the dark points are where that neuron was very active, and the white points are where it was not active. Over time, activity fluctuates. And also across populations of neurons, there are really strong correlations, and those correlations can change over time. So if you think about, like, what would you have thought the population, the entire, you know, cohort of neurons was doing if you only had one of these rows versus what can you see when you have, you know, thousands of them? It's really a different game. But this isn't the only kind of data. Oh, sorry. Yes, this isn't the only kind of data. We can also do imaging data. And this is the previous one was showing electrophysiology. So using very, very dense electrodes to sample essentially the extracellular signatures of spiking in individual neurons. Now that gives you very precise temporal information, but you don't always know exactly how neurons are laid out in space. There's another way of looking at neural activity, which is based on imaging calcium dynamics. And with this method, your temporal resolution is lower and you have an indirect readout of neural activity, but you have a lot more information about spatial positions of cells and how they're organized. So these are really complementary depending on what you're looking at. One may be more useful than the other. There are other kinds of data about neural systems, and two that I want to highlight. Using sequencing methods, we now know far more about the actual cell types that make up the brain. These are visualizations of patterns of gene expression within individual neurons, and they're clustered based on similarities. So where you see clusters, that means that, you know, there's several hundred or maybe several thousand neurons that had really similar patterns. Here, just to highlight one, all the blue ones are neurons that give like excitatory inputs to the neurons that they connect to. They're, so those, we call them excitatory neurons because if you receive an input from them, if a neuron receives an input from one, it becomes more likely to fire. The pink ones are inhibitory. So broadly, by looking at these patterns of expression, you can say, well, it's excitatory or inhibitory, but you can also see there's a lot of diversity within those classes. And really knowing which of these details matter for understanding the activity patterns I showed you on the previous slide is a non-trivial problem. You know, the third, and then the third kind of activity is, you know, you have the components, you have the observations, and we're also starting to learn far more about how individual neurons are connected to each other. Now, the scale that we have this data on is still quite small, right? Like when I last updated this, so it's probably larger now, but we're looking at like far less than a cubic millimeter of brain tissue where you can actually say like this neuron's connected to that neuron is connected to that neuron. So when you look at, say, let's see, a network model, like we don't really know how to connect them up. We also don't know what features of these sorts of networks really control the dynamical patterns we see here. So these are the kinds of questions that we work on. You know, when we observe structure in neural activity, what does that tell us about structure in networks? And we approach these really using modeling and data analysis. So that is just a very broad overview of the sorts of questions in our lab. One of the angles that we take is by really focusing on neural activity and trying to understand like what are the properties of large-scale neural systems that you could actually identify from individual to individual or from one animal to a different, you know, across different species or across different brain areas. Like what is the right language really to describe dynamics of neural activity? So really, it's not, it might be overstating it, but it's not quite overstating it. One of the only theoretical principles we really have that operates on that high level is the idea of criticality in neural dynamics. And so let me dig into what I mean there. The idea is that the brain works at in some special regime between really, really ordered, where nothing would change and it can't do anything, and really, really noisy, where you can't represent information, things don't, you know, hold still long enough to actually do anything. But in between these regimes, you can get correlations that, you know, span across networks or dynamics that evolve in a predictable fashion, at least for some amount of time. Usually to get this, you need some sort of fine-tuning or balance. Now, I'm trained as a physicist, and these ideas really come from a lot of physicists who are working in neuroscience. I think like there's the canonical example of criticality that physicists use is by looking at an Ising model. And let me just show you some snapshots because I think it'll clarify what I mean here. So in this very simple model, you could imagine instead of neurons, or well, you know, imagine your neurons at each point in space. There are neighbors, and with that neighbor, if you have a positive interaction, you tend to want to align the same way. If you have a negative interaction, then you don't want to be doing the same thing at the same time. And depending on how the interaction between neurons or sites is organized, and depending on, you know, really what the temperature of the system, like how do those energy differences matter a lot or do they matter very little, you can get very different phases of activity. So in the case where you want to align with your neighbor and the cost of flipping is significant compared to like your available energy, this is what we would call like low temperature, and you may get domains where everything is just kind of fixed. And it's very, very stable and you can't change things. Because, you know, flipping So in this picture, this is like hundreds of pixels representing these sites that interact only with their neighbors. If you flipped one of these from black to white, it would incur a big change in energy that would be large compared to the temperature. So in this, it's too static to be interesting. At the other extreme, you could flip the energy, but there's so much available energy, it really doesn't matter. And here there's a lot of, there's noise, the correlation length is very, very low. But somewhere in between these two regimes, you have a critical temperature where you'll have domains that tend to, align with each other, but they can fluctuate. They'll move around. If you look at correlations across this regime or the whole field of view, you can have correlations that span a very large distance. You can have communication in some sense between areas. And these are taken from a really fun online widget where you can play with the temperature, you can play with the grid size, you can see what happens if your system is really small or really big. So that is, I think, a fun thing to do. All right,
So correlations are
yes.
very low in both cases, the
Yeah.
extreme cases, and pretty high and expansive in the middle.
Yes, yeah. And so I think this is something that I have sort of struggled with the concept of criticality, in that how else could a brain work, right? Like your brain has to have interactions across. But so the question is like, well, how do you set up a system to get these kinds of signatures, these kinds of relationships?
And the middle one can span the whole brain.
Yeah, it could, yeah. Okay, so one signature, so when you're actually looking at a neural system, how would you know that it's critical or not? One signature may be that certain statistics have power law behavior. And I will show you more specifically what I mean by that. But there have been many different kinds of power laws reported in neural activity, starting with Zipf's law, which next slide is telling you what Zipf's law is if you don't know it already. As well, looking at the structure of covariance matrices at different scales. And then the dynamical criticality of avalanche statistics, which really does connect to physical avalanches. This is why it is called avalanches in neural activity. It's the same theoretical framework. All right, so each of these have been observed in neural activity, and one of the questions is, how is it that a brain would set itself up to actually achieve these sorts of phenomenon? Is it really that we have multiple kinds of criticality and we really need like a mechanism to understand each of them? Or maybe there is kind of one generic mechanism that you would expect all three of these different kinds. So we're going to explore that. All right, so Zipf's law is a really bizarre phenomenon that was first reported by Zipf. Actually, I think there was somebody who was concurrent with him and his name didn't get attached to it. So it's unfortunate. But so Zipf was looking at word distributions, and he found that the most common words, if you ranked words from the most common to the least common and looked at how often they appeared, that essentially the probability of an item was inversely proportional to its rank. Now, this shows up in word distributions. It shows up in sizes of cities, in firm sizes, in receipt tallies, in
How do you compute the rank?
How do you compute the rank?
Yeah,
So you have, 1,000 words.
yeah.
The one that shows up the most often is rank 1, second most often is rank 2, third most is rank 3. So
First digits as well.
in digits as well, in the next word distribution in ChatGPT, which is, there's like a part where it's not Zipfian, but then the tail is Zipfian. And then in neural activity. So this is, I mean, this is really bizarre. It's to see something across so many different systems. But there were a few papers, at least in the context of neuroscience and possibly in other fields that I'm not aware of, but that showed that you could get Zipf's law if your data actually has some latent variables. And this example comes from the Aitchison, Karate, and Latham paper, where they were looking at the distribution of words in a corpus of text. And if you broke it down by adjectives and adverbs, by part of speech, sorry, these are quite small, none of those have Zipfian distributions. They kind of have characteristic distributions for that part of speech. But taken together, when you are looking at, you know, an entire corpus of text, you're summing up over all of these distributions, and it approaches this.
So what is a latent variable?
So in this case, a latent variable is something that you don't, when you computed the first distribution, you didn't take into account. So it's something that maybe controls an aspect of the distribution. For example, you could have had, let's say you had 3 or 4 exponential distributions that have different, you know, decay constants. Then that latent variable, you might have said, okay, so that you have different decay constants in each of these cases. So here, it's kind of hard to read because they're all smashed on top of each other, but each of those parts of speech has a distribution that's slightly different from the other one. So in that case, the latent would be, you know, this is the noun distribution or something.
So it requires infinite exponential.
It requires.
In the limit, we have this law, it requires infinite number of lambdas.
So if you actually have a power law, yes, but if your threshold is a power law within the resolution that your data
Then
can
it's okay.
support, then, and that, I mean, this is also true in the neural activity. So in all of the cases where we're looking at power laws, we have a finite number of neurons, we have a finite length of time, and those cutoffs will, you know, prevent us from actually determining if there's a true.
When we prove that theorem with the exponentials, this article does it prove? It's okay.
No, so, okay, so the Schwab paper has a very nice discussion of it, and I think they get, you know, precisely the Zipfian exponent of 1 in the proof, and that's in Physical Review Letters. The Aitchison paper is nice in that it makes things very, very concrete, and it's not quite as restricted as the Schwab paper.
Thank you.
Yeah. Okay, so so the idea here that, you know, perhaps we can understand Zipf's law in terms of latent variables. What about those other forms of criticality that we see in neural activity? So with the time remaining, we're going to look at examples of those 2 and see how far we get. Okay, so I was telling you a little bit about 2-photon imaging in large populations of neurons. What these authors did is they took recordings from a mouse that was running back and forth in a linear track, actually in a virtual linear track on a ball, but it, you know, doesn't, for the purposes of the analysis, it doesn't matter too much. And from these, you know, 2,000 neurons in one brain area, the question is, what are the things that scale with the size that you analyze the system at? And the reason you might expect to find something here is that if the system is in some sense critical, then at different scales, you should see similar structures. So we see similar structure at different scales? The way they approach this is using a coarse graining analysis where they look at single neurons, and then based on how correlated pairs of neurons are, they merge them into a cluster variable. So at the next level, they have pairs of neurons that have been merged from highly correlated single neurons, and then they repeat the process. So then pairs of neurons that are highly correlated, those get merged into the next variable, and so on. And then they're going to look at how different statistics scale with cluster size. And so the first thing, if you look at activity variance, so this is the variance of the activity within a cluster, this is on a log scale. So the straight line indicates that there is a power law, at least, and I'm very sorry for the small thing, but it goes from 1 to 100, so it's about 2 decades of scaling there in the variance. Now these dashed lines are showing this is what you would expect if everything was independent, and this is what you would expect if everything was identical. So it's, you know, in between everything's independent and everything is 100% correlated. All right, another analysis, you can look at each of these clusters and calculate what is the eigenvalue spectrum in that cluster. And the eigenvalues of the cluster covariance matrix scale with the cluster size. Sorry, the eigenvalues scale with the rank over the scaled rank. So that's a couple of scalings there. And if you look at the eigenvalue spectrum from clusters of different size, they collapse onto the same, like really onto the same spectrum. This is only about 1.5 decades of scaling, but you run into finite size effects pretty quickly. I think they only went up to like 128 out of the 2000 cells. And then finally, you can look at how correlated the cluster activity is over time. And the autocorrelation time of these clusters also increases with the cluster size. So as your clusters get larger and larger, the effective time constant of that activity gets a little bit longer. All right, so in this paper, they looked at whether standard network models that we typically use to model neural systems could account for these, and they didn't. So they looked at random firing rate networks and a certain balanced excitatory inhibitory spiking network, and neither of those could generate these sorts of scaling phenomena. So working with Ilya Nemenman at Emory and a really talented undergraduate, Mia Morel, we decided to take a different approach to modeling this. And the idea was, can we put latent variables into this model and see, like, could this be a consequence of dynamical latent variables? So the is pretty simple. These are neurons in the hippocampus, which are known to have a certain selectivity to where the animal is in space. So we incorporated that into our model. We also added latent dynamics. So there were just randomly changing latent variables that neurons are coupled to. These might represent things like inputs from other brain areas, neuromodulation, and so on. The coupling to the latent fields is random and widespread, and we tune the network so that the distribution of the population firing activity is in the right regime for neural activity, which tends to be fairly low firing with non-zero but fairly weak correlations. So just mathematically, and it's always fun because there was a version of this with errors in the equation, and hopefully I got them all out, but then they propagate across slides. So mathematically, we can write down an energy for a particular spin configuration or population activity configuration, conditioned on these latent variables, and X here is just the position of the mouse in the simulation. Here, the latent variables are really just an autoregressive random walk process, and they have a fixed time scale tau, which we didn't change, and it's identical for each of the latent variables. And then we simply, as these latent variables change over time, we and the simulated mouse runs around in its track, we just sample activity and then generate, a simulated data set like the recorded data set and apply the same analyses.
Is the system out of equilibrium or in equilibrium here?
I mean, it's like, I guess it's the latent variables are changing from moment to moment.
Yeah.
So I guess that would be out of equilibrium. I mean, we're just sampling
Okay.
from the model.
From the model. No, but the probability there, no, is based on the.
Based on like a
right?
Boltzmann kind of equilibrium equation.
Maybe that is quasi-static, no?
It is quasi-static, yeah. So the time step is, the tau is maybe like 100 to 1,000.
Which is very long.
It's fairly long. It's
Enough for the neuron to get to do some kind of.
And actually, oh, you know what, the next slide is, this is like a sample of activity from the network. So, it doesn't look precisely like the activity patterns we saw at the beginning. I mean, you could cluster them. Maybe you can see similarities, right? Like there's groups of neurons, but it is a little bit less, it's maybe more realistic than a totally random network. I think there are still features of neural activity that it's not capturing, but let's see how far this takes us. So applying the same analyses from the neuroscience paper, we looked at the activity variance. Here, the model exponent here, and the data is in blue, the model's in black. So we get quite close to what was shown in the data. We look at the eigenvalue spectrum, again, really within the experimental error bars here. And the time scaling was trickier, and you see like these exponents are different. Now, if we allowed our latent variables to have different time constants, we could get much closer. So maybe that's part of the solution, but essentially, this system that has coupling to latent dynamical variables can also generate these signatures of scaling across different scales in large populations. What parameters in the model really mattered? So it was a combination, the model combined this place-dependent activity pattern as well as the latent variables. It turns out that the place fields didn't matter at all. And as long as we had more than about 5 or so latent variables, we were getting in the regime of the exponents that were found in the data. So it turns out that this is not just a feature of this one data set from hippocampus. So since that first paper, another paper was published using the same analysis, now looking at recordings from really diverse brain areas. This is, I won't step through them all, but essentially this is the eigenvalue scaling across 16 different brain areas, including subcortical and cortical areas. One thing to point out is their hippocampus recording here is actually getting quite close to the exponent that was obtained in the imaging, which I think is reassuring, because these are really different ways of recording the activity, but it's the same data. I mean, not literally the same data, but the same brain area. So maybe there's some consistency in this metric. It's a useful way of looking at, complex brain activity. One thing to note is that these actually have quite different scaling relationships. If you look at the actual exponents, they vary from like 0.5 to 1. And from our model, we can actually guess that, this could be related to the dimensionality or the number of latent variables that went into the model. It also depends to some extent on the other parameters of the model. So really figuring this out isn't quite done yet. So to kind of summarize where we are, so far we've seen that of these different signatures of criticality that have been reported in neural data, it was already known that Zipf's law could arise from latent variables. We showed that, you can also get scaling and covariance eigenvalues and cluster variance and autocorrelation timescales with this coarse graining analysis from a system that's coupled to latent variables. But maybe the real canonical example of criticality and neural activity is the avalanche dynamics. And so with the last part, we're just going to step through another recent paper that we had looking at latent variables in that system. So should say part 3 probably. Okay. All right, so over 20 years ago, John Beggs and Dietmar Plenz were recording in multi-electrode arrays and cultured neurons, and they analyzed the activity by looking at these. If you looked over time, there'd be big bursts of activity, and then it would be silent. And they found that if you looked at these patterns of activity, you could look at their total size and their total duration. And these sizes of events were power law distributed. In fact, you could, if you looked at the distribution of sizes, the distribution of durations, those gave you 2 exponents or 2 power law relationships. And from that, you could predict through what we call a crackling relationship, what they call the crackling relationship, how the average size scales with the duration of an avalanche. So in the 20 years since then, there have been many, many studies. This is a tiny sample of them, looking at neural activity in different systems, from zebrafish to mice to turtles to also like human EEG, LFP recordings. A whole number have looked for these same signatures of avalanche criticality. And in many systems, they find, again, power law, size distributions, duration distributions, and the crackling. So it's also known that you can get power law distributions with having latent variables. And that was shown in these 2 papers. But in the previous models people had used, they could not get the crackling relationship. So they could get that sizes had a power law distribution, but if you looked at the average size as a function of duration, it had nothing to do with the exponents for the size and the duration. All right, so I probably wouldn't be talking about this if we didn't see this behavior in our model, but we do. So the way that we typically look at this, we look at the distribution of sizes, that and use a maximum likelihood estimator to get the best power law estimate here. We can also look at the duration distribution and then try to see how well the tau and the alpha from the size and the duration distribution predict the size with duration scaling. And in this particular set of parameters, it matches extremely closely. Now, these exponents aren't quite the same as those reported in all of the experimental papers, but they are the same as some of the ones in the experimental papers. So really understanding the details of why you observe which one in which system is still a little undetermined. All right, so in the system, if you recall, we had, for the coarse graining scaling, we needed to have like 5 latent variables for it to work out, but we could have looked at a system with just 1. If we look at a single latent variable, it actually begins to show these really bad deviations from power law, especially at large sizes. The duration now, compared to the precision you can do experimental data with versus modeling, because we can simulate forever, like this may look like a straight line, but it is definitively not a power law. We get very poor fits with a single latent variable. And if you try to look at the crackling relationship, the predicted exponent here does not match what the actual observed is. If we take the same sort of temporal dynamics, but now just add multiple latent variables, these show power law scaling over a much larger range, and we regain the crackling relationship between exponents. And in fact, we can look at really what's the interdependence of, you know, time scale and crackling. And if you only have one latent variable, you will eventually get crackling if you make that variable very, very, very slow. I mean, essentially, eventually we were doing like quasi-static simulation where you just fix it for a very long time and then change it and fix it again and change it and fix it again. You will get crackling, but if you have multiple latent variables, you get crackling at faster timescales. And those faster timescales are probably more relevant to what neural activity is actually doing because your brain activity does change spontaneously. So essentially, this one model, and when I say one model, I mean like this idea that having latent variables in your model of neural dynamics really could account for 3 different kinds of criticality. And this crucial ingredient of having latent structure in the data, I think it doesn't mean that like that we're not seeing criticality or, you know, find a careful balancing of dynamics in neural systems. But what it means is we need to understand like, where is this latent structure coming from? It could be coming from recurrence in local networks that generate some emergent activity pattern from that population. It could come from feed-forward input from other brain areas, or shared modulation of neurons in a network. And really in a system where you're able to read out information about these latent variables, that is when we expect to see criticality, power law-like signatures. So in our model, we can also, because you can change like, what is the kind of sensor you have? Like, are you looking at imaging data or are you looking at spiking data? Are you looking at neurons that have this distribution of firing rates or that distribution? We can actually use this to predict when we should see signatures of criticality and when not. And that's an area that we're working in now to try to explain, like, why is it that we see different exponents across different brain areas? Is it something that's fundamental to the function in that particular brain area? Or could it be something more prosaic, like the neurons you happen to sample just happen to have a different time scale or distribution of firing rate or something like that? And with that, I mean, I have some more details that I went through much faster than I thought I would. But that is what I have. And thank you for your attention. And
thank you. Virtual friends, do you have questions for Audrey, Pedrak, and all others? Do you have questions? I see 12 people there.
I can.
Yeah, can you?
You know, the comment would be that exponents seem to go between what in dynamical systems is called ballistic to random walk, you know, between 1 and 2. This is a huge range.
Yes.
And when they're so dramatic, you might be able to go to a particular region of the brain and explain why this guy is a random walk and the other one is totally locked, you know, right? I mean, 0.71 is the most favorite exponent for people who are extracting scalings in this kind of subject. But you have a huge range, so that must mean something.
Yeah, and so do you mean branch? Let me pull it up.
Maybe we need to unpin you.
Oh,
I can do it from here, actually.
um,
Oh, hey, oh,
no, I
ah, you
think,
wanna go back? Yeah.
yeah.
And I showed this figure, right? So everything looks the same unless you look at the numbers. Numbers really go from
Yeah.
0.9 for 3P to 0.52 for LS. That's huge, right, for exponents.
Yeah. Yeah.
So it feels like you should be able to use this as diagnostics for what kind of neuronal activity you're having. Different,
Yeah.
you can turn it around. Maybe.
Well, and I mean, something that's kind of interesting, this is thalamic area. This is also thalamic. This is also thalamic. So is this one. And this is also thalamic. So, I mean, of the ones that are closer to 0.5, many of them are not cortical areas. Now, it could also be that you're like, these are very deep. Let me see if we have the.
Because can you explain us the difference between ballistic, what was the other, what was the other things Petra
Ballistic
says?
or random walk.
When you have in diffusion, when you have critical exponent, random walk is 1/2. And if you just go straight without changing your direction, that's called ballistic.
And in terms of brains,
Exponent is kind of
in terms
one.
of neurodynamics?
So here, okay, so on this variance plot, so if this were the square root, then this would be 1/2 and that would be 1.
Mhm.
But it would be that as you add more things, the variance goes with,
Random walk,
so the random walk is down here.
okay.
And then the ballistic is.
The ballistic is more ordered,
Mhm.
Totally.
Things going in the same direction.
in the same direction,
Yeah.
okay.
Yeah.
And instead, the one with chaos will be the completely uncorrelated and
Well, with
Well, no,
noise,
in
totally
between
random.
you have
other random
correlations,
walk,
right?
okay.
In between you have correlations. Yeah. In Mia put together, Mia Morel, the first author here, she made like a 60-page supplement to the physical review letter
Wow.
that has parameter scans in different, well, not exhaustively, but a lot of line searches in different directions. So it's interesting, what really like you can find different, like alpha and mu are tightly tied to each other. Although I don't know how close, if we looked at that in as deeply as we looked at other things. So.
And how do you relate this to Levy flights? Are these Levy flights? Levy flights?
Yeah,
Because they are power laws,
yeah,
right? So they are all Levy flights, so the variance is huge.
Okay, so I can't answer the question on levy flights, but the variance is still below, if they were all below the ballistic.
The ballistic, oh, yeah.
So, okay, yeah, I mean, in our model, there's no levy flight.
There's no Levy flight, basically, okay. Yeah, the variance doesn't go infinite.
It
To be a
doesn't,
Levy flight,
yeah,
it should
and
be
so-
infinite. This is not the case, basically.
No, I mean, not in the model. I mean, who knows what the actual brain is doing?
Oh, yeah. Any more questions from the virtual world? Evangelos, Bojun, GI, Hanglu, Sarah. Okay. What about from the audience here? Yesterday, I was in your class and I saw all your dynamics. You saw me all those spikes.
Turn to them, yes.
And have you seen the relation here? Did you see those? Can you show them the first picture when you do the plots of the neurons? Here we go.
Like this one?
Yeah.
Okay.
If you take one slide along X, that's his time would be, which is it will give you the same plot like I saw yesterday. Do you see the relation?
Say yes.
But have you seen her model? Did you see her model? Does he relate to your cost function in?
Yes, yes.
It does.
If you transform it a little bit.
Yeah.
They do
But-
water
So
resources
they-
now.
that's very, very interesting. Sorry, but that's really interesting stuff. The behavior of criticality in other systems actually emerges as an average of, or when the exponents of the exponential probability distribution are averaged over a certain time frame. Is that concept anywhere here? I mean, for example, I don't know if you're familiar with the Zipf's law there's something else called the Bedford's law for probabilities. And
No, I don't, yeah, tell me more.
so the first digit that you mentioned
Okay,
is really this. I mean, if you take any
Yeah.
number of data which are significant and you look at the first digit, then they're all distributed, they're independent. They're distributed according to this Bedford's law of probabilities from, the first, second, third, and it's always happens no matter what the system is. So in there to prove this, which I have a little proof of that. You have to average the exponents of the exponentials over the period, the periodicity of the cycle. Does that resonate with you in this case?
so potentially, so when we had, let me just-
Oh, by the way, there was a professor in mathematics here at Georgia Tech 20 years ago that also contributed to this law you're talking about, and he used it to analyze some
It's
data from the business of
So
it.
this is maybe the closest that I can get to showing you like an actual slide. Now, this is the number of latent variables that went into the model. Sorry.
there.
Yeah, over here, the NF. So the number of latent variables, and I will say about this point at minus 1.75, you can pull up the supplement and see that like it is a very generous attempt at finding an exponent because the covariance scaling, so this one is just about covariance scaling. It doesn't, it's not a very good scaling. I think by, 2 or 3, it starts to look a little bit flatter, but we really needed to have multiple in here to get like eigenvalue scaling in the covariance matrix. So I suspect this could be like underlying it, but yeah, it would be nice to have
Like to talk to you about later.
I would-
But
yeah,
the
great.
bigger question I have is that, so you're suggesting that the brain works in a way that whenever something interesting happens, there's criticality involved in terms of either thinking or perhaps.
Even with creativity, even Leonardo would do this basically, would go ballistic or would go noisy.
maybe I pulled out this slide too. So another thing that we did in the in the avalanche model. So these are the two, we're looking in the quasi-static limit where things are changing very slowly. So you only need one latent variable and you get avalanche scaling. But there's still two parameters, the gain on the inputs, and then this epsilon, which is kind of like a bias towards or against firing. So across this plane, you get very different distributions of activity. Essentially, this is how far off you are with the crackling relationship across that plane. And you're quite close. I mean, as close as any of the experiments are, regardless of what the scaling and the epsilon were. But we can also look at how much information do you have about the latent variable across this plane. And depending on how it, you know, in some areas you have very high information about what the latent variable is, and some areas you have very low information. And where you have a lot of information is also where you don't have to observe the system for as long to get the critical exponents. So it's kind of not quite as clear cut as if you have criticality, then you have good information transmission. For this model, it seemed to be like you need more or fewer observations to see it. If that does that.
And the observation would be the stress of
Like,
outside the
the
world.
time that you observe the system and how much those latent variables change. So.
And how would you relate it to creativity?
Yeah, so I don't know about creativity.
Okay, yeah, me neither.
No
Yeah,
idea.
me neither.
But
Just
if,
something.
you know, if creativity is generating some, you know, if an idea is essentially a latent variable in your brain somehow, in some incarnation, right? You have a balance between having information about lots of different ideas or or having too many ideas to really have information about them, right? Like, let's say we put 100 latent variables in there, your ability to extract information about particular ones is going to be diminished, right? But there's, you know, there's probably some sweet spot where if you have multiple latent variables, you continue to get a good return. I haven't done any of these calculations.
No,
I'm speculating.
I'm just kidding. Yeah. Can you explain more why the idea can be modeled as a latent variable?
No,
Do you have-
no, I mean, I don't think she said that. Did you say that?
I mean,
So the question is the idea or the thought, the critical organization of the brain function, or is it one thing that you're thinking? I don't think so. It must be the organization of in some certain way of a bunch of neurons and stuff. But
yeah, so I think if you have certain network structure, you will get from the dynamics of that network, some low dimensional, not low dimensional is kind of the wrong word, but like coherent patterns that show up. You may need like nonlinear dimensionality reduction to like really, so like if you looked at it with PCA, it wouldn't look that low dimensional, but that there's some, you know, structure.
Some emerging structure.
Yeah.
And that would be the idea,
And,
basically.
you know, I mean, Chaim Soplinsky and, I forgot the first author's name, but they had a nice paper on, the dimensionality of activity in random networks a couple of years ago. So like just having a big heterogeneous network with a lot of variability and connections between neurons, you can get these sorts of emergent latent variables. So then you could describe maybe a mode of that network activity in terms of one of these latents. Perhaps.
No, I mean, it's
Yeah.
really interesting to explore it.
Yeah.
You have the model, so
Yeah,
you can
It would,
test.
and so I think that what would be really interesting is to approach this actually from the dynamics side and like when you have some, you know, random network that is modeling brain activity somewhere, right? Like, how do you go back and forth between these two views? Like, can you approximate it using the statistical model, and then really learn more about like the dynamical phase space or something?
And then, of course, the human brain is a bit different than, it's organized, I think, in layers, isn't it? I mean, it's like.
So that's a great question, because these are, I mean, most brains also have layers.
Yeah.
Their layers are different, their cell types are different, the organization is probably slightly more elaborated, but one of the things that wasn't done in that really hasn't been done. Now, if you look at these, I'm sorry that it's very small. And I'm not sure if I can actually zoom in, but, the cortical areas do have layers, and if you imagine you're sampling them with an electrode, that electrode might be going straight up and down, it might be going sideways, it might be going across a layer. So I think really knowing what the geometry of the recording was and what particular population you're sampling is important for interpreting these recordings, right? Like, I mean, you would need the electrode tracks to know if the differences that you see, say, between primary visual cortex and, oh, now everything looks the same as primary visual cortex. And let's see, I don't know, I think this is orbital frontal cortex. Like, is this difference because one of them is more frontal, one of them is more posterior, or is it because you happen to be sampling a totally different layer in one brain area than the other? Sorry. Oh, someone had a question.
Edrak, did you have a question? No, it was just,
Okay.
So on this, just one more thing about this one. So in this, in all these possibilities there's no clustering. You say plus or minus this and that, but is there clustering within those lines? Because that could indicate what you just said, that the feedback or whatever the learning variables come from different layers, which you know, are translated into this ensemble of curves. But are the curves clustering somehow? There's a distinction.
You mean like if you looked at these all together on the same plot, would you see?
Right, what do you see? Do they? Yeah.
And they didn't
indicate that if
Yeah.
if it happened.
Yeah.
Sorry.
okay, more questions? Okay, thank you very much, Audrey. And let's go and get the pizza downstairs because Audrey has to teach at 2:00.
I have to teach that too, yeah.
So, hey, worst case scenario, you take a
I
pizza.
take it with me.
So
It's great.
get you back.
Good to see you.
To tell us
How this goes?
how this works and why are we the way we are.
Thank you.
Thank you, Pedro. Thank you.